Mitosis Land by Samantha
and Deanna. What is mitosis?
Mitosis is a type of cell division that results in two daughter cells.
There's not one, not two, but 3 reasons cells divide. The first reason is reproduction. The second reason is repair. The third and final reason is growth. These are 3 reasons why cells divide in multicellular organisms such as cats. In unicellular organisms such as eggs, there's only one reason why cells divide. This reason is reproduction. Unicellular organisms do not grow and do not repair because there is only one cell.
In mitosis, there are 5 phases. The first stage is interphase, or between phase. The cell spends most of its life here. Interphase is broken down into 3 stages. Before G1, growth or gap 1, DNA is in a loose state called chromatin. These are chromosome Twizzlers that are arranged in pairs. Chromosomes are complexes of equal parts DNA and protein. All humans have 46 chromosomes that are arranged in 23 pairs. This picture is called a karyotype. Before the cell can divide, DNA has to be copied. The parental DNA double helix is unwound, and new DNA strands are synthesized.
During S phase, or DNA synthase, chromosomes are duplicated into sister chromatids. Sister chromatids are exact copies of each other. The chromatids are held together at the centromere, or the musketeer candy bar.
At the end of interphase, there are duplicated chromosomes that are in a relaxed state and duplicated centrioles that are in clusters.
The second phase of mitosis is called prophase.
In the beginning of prophase, the nuclear membrane breaks down and DNA begins to condense into chromosomes. Centrosomes move to the opposite poles of the cell and spindle fibers form. At the end of prophase, DNA is fully condensed and the nuclear membrane is totally gone. The chromosomes are attached to the spindle fibers.
The third phase of mitosis is called metaphase. In metaphase, chromosomes are aligned along the center of the cell by spindle fibers.
Anaphase is the 4th phase of mitosis. During anaphase, the spindle fibers start to shorten as the sister chromatids are pulled apart.
Telophase is the final phase of mitosis.
The chromatids reach the poles of the cell, and in the middle the cell starts to pinch. The events of prophase are reversed, and DNA decondenses, the spindle fibers disappear, and the nuclear membrane reforms.
The cell cycle is controlled by checkpoints. If a cell does not correctly fulfill each checkpoint's requirements, the cell cycle is paused. This problem can be fixed or the cell can eliminate itself. There's not 1, not 2, but 3 checkpoints the cell has to go through in the cell cycle. The first checkpoint is between G1 and S. The second checkpoint is between G2 and M. The third and final checkpoint is between metaphase and anaphase.
Stop! Is your DNA intact? Have you completed DNA replication? All your chromosomes aligned at the equator?
These checkpoints are controlled by the activity of CDK proteins. CDK stands for cyclin-dependent kinases. These CDKs are always present in the cell. There's a growth factor that binds to receptor and stimulates cyclin synthesis. The cyclin activates the CDK, the cyclin-dependent kinase, and the active CDK stimulates DNA replication in the cell.
Cyclins are present at a certain time during the cell cycle and then disappear after they've done their job.
There's 4 different cyclins: cyclin A, cyclin B, cyclin D, and cyclin E.
Cyclins activate CDKs. In this demonstration, when you plug in the charger, it activates the phone to start charging.
Active CDK cyclin complexes phosphorylate key control elements like RB into the major control protein PRB. One consequence of control mechanism errors is oncogenes.
Oncogenes speed up the cell cycle, activating mutations, which leads to cancer.
The other consequence is mutated tumor suppressor genes.
Tumor suppressor genes slow down the cell cycle, deactivating mutations, which also leads to cancer.
Thanks for watching our presentation on mitosis.
Arrivederci!